genotype. If I say phenotype, I mean how it looks. Tall, short, white, black, and that's the phenotype. Pheno means physical, gene means genotype. And they come either, you always got two letters to a set. They can be two big letters, two small letters, or one each. Now if you're two big letters, you're homozygous, dominant. If you're two small letters, you're homozygous, recessive. If you're one each, we just say you're heterozygous. Because it's understood you're dominant. You ain't got to say dominant. That's understood. Because if you're like this, the big P is going to be the one to take over and cover him up. All right, now, next slide.
Sorry.
Hey, listen, I got paper for you.
I'm sorry.
Paper for you. Do you know what it means to be segregated? What's it mean?
Separated.
White, black. Separate, right? Boys and girls. I know we used to do a lot of segregation where it's 6 classes. All the boys were in one class. Girls in one class, they can't embarrass each other. A lot of guys are shy and they have questions, but they want to ask them, the girl's sitting there. Girls don't tend to care. They'll ask questions regardless, but boys, they don't volunteer anything. And they might even do it with a bunch of guys because they might lose face. But segregation means to separate. And he had an idea about separation. The alleles, these are the P's, big or small, they separate, they segregate. And you're going to find down here at the bottom, this is a sperm. It contains this one. This sperm contains this one. But they're separated. Each went into a sperm. And the same thing here is going to happen. It's the same letters, so the two eggs, and I got P. I guess I used my stereotype. I'm using pink for a female and blue for male because you couldn't hardly see yellow, so I used blue instead. But obviously, the two eggs produced can only hold the same allele. And most folks just write it one time, realizing it's the same one. But if this is the male, this would be the pollen. And all the pollens is exactly the same. It's all big P. If this is the female, this is the egg, that's down there in the flower. And they're all the same. So, no matter how you do it, if this one finds this one, it's a big P, small p. If this one finds this one, big P, small p, this one and this one, it's the same thing. Now, there's a thing we use. You ever hear of something called a Punnett square? I don't even know, I should know his first name. Punnett was a man. And I just showed you how to go this one, this one, this one, this one, this one, this one. But he has a square he does. And he puts a square on there. And in the square, he combines them. He takes this big P and puts it here, and small p and puts it here. And he says that this individual will be big P, small p. Now what color is that going to be? It's going to be purple. So the genotype would be heterozygous. The phenotype would be purple. And over here, this one and that one, well, when you fill the box in, you get this. They're all going to be exactly the same. Now how you pair them, all 4 are heterozygous, big P, small p. And they're all purple flower. Now notice that the white gene is there. This is going to be a white flower now. He's there, but he's being masked by the dominant allele. And that's why, I'll tell you right now, this one each is just as purple as a double big P. You can't tell by looking if he's a heterozygous purple or homo, because they ain't no difference in the shade. Because this is complete dominance. Now if you're a rose, you might get a lighter pink color because it's a blending effect. Now if you get a blending, that's non-Mendelian. If you're Mendelian, then you have either or. If you have anything dominant, you're going to see that sucker show up in your offspring. And to be recessive, you cannot have any dominant gene, because if it's dominant, you lose the recessive trail. So that's a Punnett square, and if I go back, from this flower came these eggs, and I said this is going to be the color of the lady. And from this flower is the pollen. We don't say sperm, but pollen is sperm. It's just in a flower we say pollen. And if this pollen finds this egg, you put it right there, big P, small p. And how you do it, you're going to get the same thing every time. All are going to be purple, all going to be heterozygous. Now, what happens if this one self-pollinates and he makes seeds and you plant those seeds and say there are 100 seeds from this flower and you plant them? You will see some whiteness then, buddy. And here's why. Let me back a minute. When these flowers came up, now remember now, he crossed a white flower and a purple flower, and he was surprised to see no white flowers. He was thinking, surely there's going to be a white flower somewhere. Never was. Because though they had the recessive trait, they had the big one also. And that made all of them purple. So Mendel was confused. And he kind of got discouraged and said, well, I'm going to stop. Well, the flowers then self-pollinated. He didn't do it. They did themselves. And then he said, well, we'll plant these things, what happens? They planted seeds. Now, the F1s, I tell you, they're all this. Now remember now, these are your F1s. These are the F1s. And they're all heterozygous. Therefore, the phenotype, purple flowers. Okay? Now what happened? Now, this one, this purple flower, now look back, it looks just like that purple flower. There's no difference except for the genotype. The phenotype is the same, purple. But this is a genotype which is totally different from that genotype. They both produce the same phenotype, they both look. But obviously the genes are totally different. But the fact I got one big P is all I need to make a purple flower. Two don't make it any darker than that one does. So the question is, what happens if these flowers pollinate each other? Now, if you do segregation, you're going to find that the big P goes into one gene and then in another one. Now, I shouldn't have made that red. You know I shouldn't have made that red?
Shouldn't have.
It's all pollen. I should not have. But that should be blue also. I was trying to show you differences, but what I should have done, if this is going to be pollen, both those should be blue. And that's going to be the egg. They both should be what color? If I keep myself straight. Can I fix that real fast? Now mine now looks different than yours, but you know what I did, right? I want both these to be pollen, so I don't want a red line around that one. And both these are going to be the egg. And now when I bring it back up, and you see, oh, I got to do one more thing. I got to take this. And I'm showing you these two alleles being segregated, separated into the two pollen grains. And I have those blue. So I got a big one in there and a small one in there. And I'm going to have these segregated also into the egg. And I have either a P or a small p. Now, if this one and this one get together, it's a big and small, right? If these two get together, it's going to be a big and big, right? If these two get together, it's going to be a what? Well, the Punnett square can show you that pretty well. So the thing about it is, the segregation tells me I got these into different. I want to show you one thing also that if we do a bigger monohybrid cross, I'm going to come back to N, where N equals the heterozygous pairs. Question, and this one, how many heterozygous pairs do you see there? One or two?
Two.
So So what is 2 to the second power equal?
4.
I got 4. Now this one, how many heterozygous pairs do you see?
1.
And what's 2 to the first power?
1. 2.
2
times 1 is 2.
So I'm looking for 2. All right. So segregation says.
So you just find how many times, how many pairs to equal that 32 of those combinations?
There's that many combinations using 1 letter each before you use 1 twice.
2N.
Yeah, 2. If there are no heterozygous pairs, like it's all homozygous dominant, then they're all, you got 1 kind. That's it, buddy.
Yeah.
All big letters.
Okay. That could take a while.
And yeah, it takes a while. That's why we rarely go over try.
That's madness.
Dihybrid with 2 crosses, well, all you want. Try would be ones that want to challenge and see how good they are. But you can make a tribal cross very simple by making all homozygous dominant. It won't matter then whether it's 3 pairs or 44. If they're all the same ones, they're all dominant, then you use it. But what I'm trying to get at, that those 2 P's, one goes into 1 gamete pollen, and one. Now, if you had 2 more letters over here, then you'd have 2 down here. 4 letters gives you 2 in here, but it wouldn't be the 2 same letters though. So I got to separate the letters out. All right, now if these got together, and to do a purebred cross, I got to decide on the letter. I got to write the genotypes. And these are things I'll be doing after the next slide. You can't do it before you see the slide. So let me show you the slide. I'm going to use tall P's. And since tall is dominant, I'll use a capital T. Because a small T does not look like an I by an L, obviously. That makes it nice. All right? So, if you segregate, now, let's say that this is the male, and we go here, and that's the female, and goes here. Now, what letter, of these 2, what letters are possible? Only big T's, right? And of this parent, what letters are possible? 1 each. Maybe the same letter. So that's all you got. From this parent, 1 can go in this egg, the other in this egg. And on this parent, 1 in this pollen. Now, in the Punnett square, we pretend that these 2 found each other. And that these 2 found each other. Now, if these 2 come together, what's going to be right here?
Big T, little t.
Heterozygous, right? How about right here?
Same thing.
Heterozygous. And right here? Heterozygous. And right here. So, tall plants, short plants, and the youngins are all what? They're all
Heterozygous.
tall. There's daddy and there's mama, and they all look like daddy does. They're all 6 feet tall. Mama's a foot tall. Daddy's 6 feet tall. Quite a bit different than those 2. But when they mate each other, and you go, see, Mendel thought surely there's a small piece somewhere in this bunch. Not a 1. Not a 1, because we now know why there's not 1. They all come out looking like that, and they're all tall. But they got the trait though to be short. But because they have 1 big T, that makes them tall. That makes them tall. 1 big.
Determine which side to put it on.
You always write the big T first.
So the capital is going to be on the left side?
Oh, it don't matter. If I put small T's here and big T's there, in here there's still, I write big T's first in here.
You still have to.
Yeah, you must write the capital letter first. So if these 2 things are over here, and those 2 up there, like I had small T's, big T's, I'm looking here. Because when you get done, you still put big T first. No matter where they came from. So I don't, my habit is to put these over here and those over there. You might reverse that habit. You might put these on top of those. It won't matter. Because if this is a small T and that's a big T, you still get the inside.
Can you have the same?
Only if these are big T's also. Because see, Daddy puts 1 T in 1 pollen and the other T in another pollen. Mama puts 1 small T in 1 egg, other small T in another egg. Now chance plays. If this pollen by chance finds this egg, there's your baby plant. if this pollen finds this egg, there's your, what if this was the egg and that was the pollen? Would it be any difference? We're still the same combination. If this were the egg and that were the pollen, it wouldn't make any difference, because you still got the same combination.
How many times before that big T and little t reproduce before it becomes TT?
The next time, when this one's allowed to mate with this one,
Yeah.
you can get your T's back, your small T's back. And the next one shows you that. So right now, I know, now pretend I didn't know this. I have tall P's. I got some short P's. And I don't know who's dominant. And I cross them. And every single one is tall. That tells me who's dominant. If they're all short, then what would be track case? Now I'd be using big S's. But since they're all tall, I pick the letter of the dominant phenotype, which is T for tall. And then for the short ones, I don't use the S, I use the other T. Either you're tall or you're the other way. And the small T means you're the other way, which we know is short. Now, if these cross themselves, it's possible that this big T might find that big T, and the youngin be two big T's, right? Could this small T by chance find that small T and get a short youngin? Yeah. Here you can, but this combination here, these two big T's will always mask those two. Because they're always going to end up being the boss. I don't care whether they're in the egg or the pollen. It does not matter.
And it will never occur that they have like some weird medium plan?
So now, the next step, we just saw that. Now, so, y'all write that down real fast. Just copy that. So you can answer I got some questions. You get a copy here?
I do, yes.
Okay, everybody got it down?
That particular.
Then just copy this one.
What's on my paper?
You got that down, right?
Yep.
Great. Now, here's my first question. What's the genotype of that purebred cross? Tell me the genotype.
Big T, little T?
Bingo. Big T, little T. Or you can say heterozygous.
That's the first reproduced.
Yeah.
Right?
If I ask for the genotype, the gene type, you can say big T, little t, but why not just say heterozygous? I know what that means. I know that means big T, small t. You follow me? So you can say big T, small t, or you can just say heterozygous. And I know what you're talking about. So they're all heterozygous, which means big T, small t. All right. Now, next question. Now describe the genotype. What are the genes?
Tall.
No, what's the actual genes? You told me first it's heterozygous. Now tell me the actual genes themselves. Now say you're big T, small t.
Big T, small.
That's the genotype. When I said describe, you can use heterozygous. But when I say give me the genotype, then you say big T, small t.
Oh,
Okay? And then I ask you, well, that's the genotype, big T, small t. That's what you write down. You don't say big, you say just two letters. So you don't need to say big T, little t. You don't do that. You just write the two letters. That's good enough. And then I ask you for the phenotype, what did you say? They're all tall. That's how you say they're all tall. They're all tall. So that's how you break it down. And when I was in college, I wasn't sure what the professor wanted. And when he asked me to do the genotype, I gave him both too. So I'd say heterozygous, and then I'd say big T, small t, in case you need to know that too. I'll give you both of them. Well, you don't let me take off 2 points from me, I'd give me everything you need to know. So I would always just say both of them. And there's no harm in them in saying too much stuff unless you're wrong. But, you know, if you ever get a question, you go, what's he want? Does he want the word heterozygous? Does he want the genotype? I mean, give me both of them. That way, you know, you covered. You know, you covered that way. Okay? Here's homework. I used to raise betas. Those are fighting. They're beautiful fish. The two males will kill each other. They have the most aggressive nature I've ever seen. Females don't have a problem. You can put females in there by the dozens. They don't bother each other. It's the males that can't. You put a mirror up there, he'll fight himself to death. That's how mean that sucker is. He'll fight himself to death. You see all these flowing fins? That's a big old body. And when mine, and I let mine fight, so when they fight, the hormones rage and the colors get prettier. If they never fight, they never get, you know, fired up. And I'm watching them, but what happened, when I put two in there by themselves, actually what I did, I kept them in the same aquarium, but they stayed in little cubicles. And they breathe air. So you got to have it exposed so he can come to the top and go up air. Because he'll drown. I learned it the hard way. He'll drown. Oh yeah.
They don't have gills?
Huh?
They don't have gills?
Oh, they have gills. But they're so active, gills can't give them the oxygen. So they'll also suck air to make a difference up.
Oh, okay.
And when they're down there fighting, just go into it. Like time, they both go up side by side. Come back down like wrestlers. And go to it again.
They'll tag out.
Yeah, and neither one takes advantage of the other one. Apparently, you know, they're gentlemen. And I don't see a signal. I just see them stop. And they both go up to the surface.
They probably both get tired at the same time.
I guess so. What's going to happen, though, how they kill each other, they're tearing fins out. You see pieces of fins floating around. Before long, they can't swim there anymore, and they drown. So the loser drowns.
Wow.
I don't, you know, beat you up by tearing you apart. I just remove your ability to go up to the very top. And when I watch them, I see them much in trouble, I take my own dad and say, oh, you get out of here. I put it back in a little cubicle. And then fins go back. Both of them go back even more vivid than they were. And it was the males who always took care of the eggs. They would make the females. You can have many females in there. And the males wouldn't bother the females, but they would mate each other and then get it back side by side. And then the male would take the fertilized egg in his mouth, and he would make bubbles in one corner of the aquarium. And you see him going, spitting the eggs up there in the bubbles. And then he would be under him, fanning them, and the next thing you know, you had baby betas. And I'd take them and sell them $5 a pop. You're making money that way real fast. Now, I'd have a variety of colors. These two are purebred here. And I haven't done anything with betas. They're called Siamese fighting. They're betas. Everybody calls them Siamese fighting fish. I guess they're from Siam. I don't know for a fact where they're from. I don't know if they live in the wild. There must be so much lake water they can separate each other and not see each other or kill each other. But in my aquarium, like I said, you can put a mirror up there, and he'll attack that glass like you wouldn't believe, and never know that the other guy's moving just like I'm moving. He's good. Yeah. But
That's funny.
so here's the homework. And, okay. And this is actually how they line up beside each other when they're duking it out. So you have yourself some Siamese fighting fish. Some are red and some are blue. You keep them in their separate aquarium. But you wonder which color, if I cross a red male with a blue female, will I get red fish or will I get blue fish? Well, I get a variation, but these are Mendelian. So you don't know which color is dominant, so what you do mate a blue fish. And I don't care if it's a blue male and a red female or a red male. It don't matter who's who. But you know, I don't care, boy or girl. Whatever color is dominant is going to be when you see the youngins. I don't care who the baby who was. So you know that red and red always give you red. And blue mate blue, always give you more blue. The question is, if I let these two mate, of course they won't mate. These are both males. But the female fish is about this size right here by herself. She's not big at all. But question is, if a red male makes a blue female or a blue male red, what color are youngins? What color are youngins gonna be? So you make the red and blue. Now here's the result. I'm gonna tell you what you find out. All the fish are blue. Every one you got is blue. The red is gone. So I got some questions for you. I want to know the homozygous dominant trait for those fish. I want to know the phenotype for the fish. For example, if you're blue, what would have had to be your homozygous genotype? If you're red, what's your homozygous genotype? And the youngins who are all blue, what would have to be their genotype?
And they're all Mendelian.
Huh?
The betas.
They're all complete.
Okay.
They're all Mendelian.
You want it right now? Or?
No, that's homework. Now, I'm trying to if I can make it any easier and even clearer right now. Oh, hang on. I want to add something to it and make it clear. I want the parents. For the parents. I want the, this is for offspring. That's the babies for the offspring. A friend of yours has a bluefish. But he's wondering, is it purebred, which means homozygous dominant, or is it a cur, or a hybrid, which is heterozygous? Which one of your fish could you make your friend's fish to, and that lets you know what that fish was? I know he's blue, but you don't know, is he purebred blue, or is he that heterozygous blue? And he asked you, because you have all these fish, if you could tell him through your mating what his fish was. Is it big B blue, or is it big B small b blue? Now you can cross that fish with one of your kind and know exactly what that sucker is by the youngins that came out of it. You following me? And that's called a test cross. I did it myself a few times. It takes a little while, because it ain't like overnight. It takes weeks for it to happen. But the question is, your friend's blue fish, is it big B blue, or is it big B small b blue? And you don't know what he is, but you can cross him with one of your fish, and then you know what he is by the youngins they produce. Which one of your fish we had to cross it with to figure out what his fish was. And I want to know why that worked. And that's a very good reason why it worked. And if you need a hint, research what they call test crosses. That might be a way you want to find out something. A test cross is what it's called when I was doing it. All right. Huh?
Okay, let me see number 4. See, I don't know how to do all that.
You got blue fish and red fish. So which one of your fish, blue or red, would you cross his blue and red so you could figure out what his blue one was?
You want to do this?
No, see, I don't
Do you
know how to
have-
take.
So you got
I don't know how
pure.
to take pictures of it on this
I'm gonna
thing.
text you. Can I text you?
Yeah.
Yeah,
Huh?
I got text message. Okay.
So you got pure. See, you have purebred blue, and you got purebred red, and all your purebred blue always make blue, and all your purebred red always make red.
Mm-hmm.
Now when you cross your two, you get always blue. Now when you cross those suckers, you get different things. But the thing about it is, your blue fish, when crossed with your red fish always give you blue fish. Now, he's got a blue fish also. He bought from PetSmart. But you don't know what it is. Is it a purebred blue, or is it a half-breed? They call hybrid blue. Does he have two big bees making them blue, or does he have a big small bee? And he asks you, would you take my fish home and cross it with your fish? And then can you tell me what, and yeah, you can, if you pick the right color fish you crossed it with. And then you look at the youngins, and that will tell you what his fish was. And when I take the homework out, if you need to, I'll explain to you how it works. It's pretty simple. It's really simple work. It's an intuition what's going to happen. There's nothing to it. But if you need some little outside jump start, look up test cross. And this is an example of a test cross. Horses, same with your horses, same way. Find whether your horse is purebred or not. Okay. I think I'm going to stop. I'm about to get into my big process. Yeah. I'm stopping right here. Because I'm about to open a new can of worms. And I think right now we've bitten off and done.
You're about to open another can of worms over there too.
Oh, we are. I thought the same thing. We're about to open a can of worms. And now, I know that Jared's going to take care of his wedding plans. Ugo's going to come in, I know, eventually. So right now, I asked Michelle, give me 2 trays in there. So that'd be 2 on one tray, 2 joined by Ugo, 3 on the other tray. So I don't know how you want to work together, but I don't have a PowerPoint plan because my instructions I prepare for y'all are just really cut and dry. Have you read those things? If you don't read them, let's go over them, do we?